Today we will be discussing about the lesions of the cervix as well as the uterus. It's the commonest malignancy from cervix, that is carcinoma cervix, and the pre-malignant lesion of carcinoma cervix, that is cervical intraepithelial neoplasia. We will be also discussing about the malignancy arising from the uterus, that is carcinoma of the endometrium. So what are the pre-malignant lesions of the cervix? It is also called the cervical intraepithelial neoplasia. Here what happens is that in the cervix there will be cellular changes of malignancy like increased mitotic activity, proliferation of the immature cells, atypia of the nucleus and increased nucleus-cytoplasmic ratio. And there are three types of CAN, CAN1, CAN2 and CAN3. When the lower one-third alone is affected, it's called a CAN1. Up to the two-third is involved, it is CAN2. The whole thickness of the cervical epithelium is involved, it is CAN3. But there won't be any invasion into the basal membrane. What are the causes of CAN and carcinoma cervix? It's said to be a viral etiology, that is human papilloma virus, and of which the 6 and 11 strains are low risk groups. The high risk groups are human papilloma virus 16 and 18. And what are the cancers produced by this virus? Carcinoma cervix, carcinoma vagina, and there are vulvar cancer. In males, it produces carcinoma of the penis. So now these are the malignancies which are prevented by vaccine. Now we have developed vaccine against the human papilloma virus, that is human papilloma virus vaccine, known under different trade names, and it can be given for adolescent girls. The government of India is recommending to give it between 11 to 13 years. 2 doses depending upon the type of vaccine. Sometimes it will be 3 doses also. So the vaccine preventable cancers are there. And coming to the screening of cervical cancer. So the conventional screening method is a Pap test. That is using, you might have seen in the OP, taking a Pap smear with an air spatula. And it will be, the patient should be coming with when she is not having periods and there should not be any infection. We will be rotating the air spatula after touching the cervix. It's a method of exfoliative cytology. The cells taken are put on a glass slide and the fixative is added and stained and looked under microscope. The another method of that is more effective is a liquid-based cytology. And in low resource settings, the other methods for screening are VIA, that is visual inspection after applying acetic acid over the cervix. So you will be seeing